doing here. Sort of like a little icebreaker to jump right into it. We thought that we would ask you to see if you could name this fossil. Feel free to write your guesses in the chat box. And I'll give you a little bit of time to do that.
Chat box.
Can I participate?
Yeah.
No, but you can answer it after.
Oh, yeah.
Is that a subtle hint?
Maybe
Maybe.
not.
Mm.
A lot of turtles.
I'm thinking that's part of that elephant husk, but
Maybe. Let's get our guesses in, and then we'll consult our expert.
Which expert are you going to consult?
All right, Jeanette, what do we have here?
That is a Gomphotherium, John. I'm assuming this is the one on the northwest side because it looks different from the one we saw on Saturday.
Mhm.
And the teeth look different. Is that an M1?
I don't
John?
know if it's an M1 or not, but that's
I
Is
can't
this
think of
the
an M.
specimen that we dug up last
Wow.
the other day? What are we?
I don't know if
looking
that's the one that's on the northwest side.
at? Oh, I see.
or
What
no.
you know your Gomphotherium children. This is
Yeah,
not
yeah.
the one from
That's
our
the Gomphotherium.
Saturday.
Oh,
that
okay.
was not, yeah, that's what I was gonna say because I actually took a picture of the one from Saturday.
Is that the one from last weekend then, Steph?
I think it's from even last season. This is a picture that
Okay.
You're looking at a right dentary, I think, right there. We
Yeah.
only exposed the
Unless this is the one that Caitlin found in fall of 2019. I don't know.
Yeah,
Wow.
that's from a while ago, but that's a cool one, right?
I know my
See
God.
the back of the jaw and then the teeth.
we figured that would be apropos to use tonight. So name that fossil. That's your Gomphotherium right there.
But the thing is, you didn't include a scale. See, that's the fatal flaw.
That's a good point. I can't plead the fifth. I didn't take the picture, so I would have thrown something down there.
Right. Could have been the size of a mouse.
That's funny.
All
All
I
right,
could
John,
think of
we're
was
gonna-
Gomphotherium because I just learned that word when I went on that field trip. I had no idea that those existed in Florida before.
Pretty cool, huh?
Most people
Yes.
don't know until they meet Jeanette.
Yeah,
That's true. Until they meet Jeanette, you wouldn't know that, right?
All
exactly.
right, John, we're going to turn the
So-
floor over to you to sort of give some background information on why did we start doing these teacher PD cohorts, taking them out to Montbrook? Why is this so important for teachers to learn this information, or however you'd like to sort of set the scene for today?
Well, I would say right. Well, I mean, the reason I started participating in these is because we had this site that we've been digging and received a lot of funding for taking care of these fossils and studying these fossils. And as part of that, we received funding to do some broader impacts. And so that was sort of the beginning of our involvement. In terms of working with teachers, the first one of these that I participated in was last year, and it was all virtual because of COVID. And but it was still really great. I thought it was a really incredibly fantastic experience, even though it was virtual. And so the part of it I think that people really got a lot out of was when we sent the matrix to them. We did sort of an overview of the site and talked about it. Then we sent them the matrix, and then they picked through the matrix. And then we met several times like this on Zoom, identifying various fossils, many of them fish actually. And then we were able to drop into various classrooms and talk about being a paleontologist or the science of paleontology, or help with identifications of fossils that the kids were looking at. So in terms of my involvement, I've sort of been learning about this process as we go personally. I mean, I know Brian and Stephanie and Bruce have done a lot of these things, and for sure Jeanette, but this, in a sense, I've been kind of learning too. And I would say that in terms of vision and sort of how this kind of experience translates to the classroom, I was really inspired by listening to all of the teachers a couple of weeks ago talking about their perspective of that, which we did when we were shooting the videos, which was really cool. So I think actually I don't even need to tell this group here why this is important, because I think they have a pretty clear view of how this kind of hands-on experience can be translated into the curriculum, whether it's just to inspire people or whether it's to synthesize information that they're trying to teach their students, through these sort of unifying concepts. They covered a lot when we were talking about this a couple of weeks ago. So I feel like this group kind of has set the vision for me, or defined it even a little bit better in terms of why this is important. It was really neat to listen to how important that was. And I will say, a couple of weekends when we did meet as a group, that, as much as I liked last year and everything that came out of that experience, being together out there at the dig site and talking about all of this together was far superior experience. So I'm really looking forward to this Paleo PD, because we kickstarted it with that experience, and I feel like it's already even a better quality experience. So anyway, I'm really looking forward to hopefully maybe dropping into a classroom or two and talking to students, and I'm also here, and I know Bruce is happy to do this kind of thing, and others. I'm here to check in if you wanted to talk about, what are the fossils that you're finding in the matrix that you're picking out, and I can help identify them. A lot of them are fairly common fossils that we find, so they're not hard to identify. Even if we just see one or two little bits, we kind of know what we're looking at. So anyway, I'm happy to do that. And I don't know what else can I say? Anyway, welcome to the virtual component of this. I feel like I've met all of you already. So and I guess with that, I'll pass it to Bruce. Bruce has a lot more experience with these kinds of professional development workshops, and understands sort of the division of how they're structured usually. So Bruce, did you want to add something?
Well, thanks, John. Yeah, I just want to explain to the teachers what broader impacts are. If you don't know, you're helping us do good science, because the funding agencies where we get the money to dig it at Montbrook, but also to steward the collections, to curate the collections once we get back into the museum, is not enough. The National Science Foundation, where we get our money to do this, also expects us to do something that reaches out to make the society a better place. And one thing we can do this by is through teacher outreach and education. So by your participation in this project, you're helping us to do the broader impact requirement of our grant proposals. So this is definitely a two-way street. You're learning from us, we're learning from you, but you're also helping us to fulfill the requirements of the funders of these projects. So thank you very much. The other thing I want to say is that last year, the teachers, now Susan, you can probably attest to this as well, because you're repeating from last year. The teachers found most of what you're going to find is stuff that we would predict. But last year, there were some rare fossils that we had never found before. John, correct me if I'm wrong. Was it some tiny lizard jaws or something that were basically new to or exceedingly rare in terms of what we knew from the site?
Yeah, I think that's right. And I can't remember whether maybe it was a part of a jaw of an amphisbaena. Does that sound right?
Yeah. So what that means is that, okay, most of what you're going to find has already been found, but then there's going to be these rare things that nobody's ever found before. So you and your class has the potential to discover species that are new to science from Montbrook, and that's really cool, because you're not just sorting the fossils, you're actually contributing to the science. And this is in a sense what's called a citizen science project, where you're contributing to science through the sorting of these fossils along with your students. All of you are part of a citizen science community, which is an important unifying concept for how we can work together as a cohort. I also echo what John says. I actually really enjoy going into the classrooms. I'm going into the classroom in Palm Beach County later this month to talk about the geologic timescale and evidence for evolution. So if you want any of us to come into the classroom and help identify your fossils, once you've aggregated some of them, we'll do our best to help out. And that's something we like to do. So I'm going to turn it back over to to Brian, I think that's all I would like to add. And okay, Brian, take it away.
And I just want to echo those statements once again. We're going to throw a lot of information at you over the next couple months. And I don't want you to feel overwhelmed, as John and Bruce said, and I'm sure Jeanette feels similarly. If you have any questions about the content being presented, please feel free to reach out. You can reach out to Stephanie or myself, and we could forward the information. Feel free to reach out, because again, we don't want you to feel overwhelmed. We want you to feel good. We want you to feel like you understand what's going on, and you feel ready to relay the information to your students about exactly what you're hearing. Whether it's just a conversation with a scientist or you want them to come to your classroom, whatever it is, please keep us in the loop so we can make sure that you are feeling great along each step of the way as we go through this professional development workshop. So to sort of close out that part, you sort of heard from us as we're framing this workshop. Now we're going to sort of turn the tables, and we want to hear from you. You just had this incredible experience that not a lot of people had. You had the opportunity to go to Montbrook. You had the opportunity to do it alongside experts and scientists and paleontologists. So we want to hear from you, knowing that you do have another couple months of this workshop. You're going to be implementing it in your classroom with your students. You're going to have a scientist come to your classroom, likely virtually at some point over the next couple months. We just want to hear from you and sort of get a general sense of how are you feeling? Do you have any questions about what we're doing? What do you want to learn more about? Do you have any reflection additional from what you shared last time about your time at Montbrook? Really, we just want to open up the floor to anything that you want to say, whether it's part of these framing questions or anything else you'd like to share. I'm just going to sort of open up the floor, and anybody that would like to unmute, please go right ahead.
I have a question. So I got the matrix. Thank you, Stephanie. Because I accidentally left it at the site because I was so excited that I left it. So I apologize. Thank you for that. My question is, how do you use it in the class? I have 130 students, so like 25 to 28 at a time. What would you recommend? Because it's a small little bag. How would you recommend I use it separated among, 5 periods of kids or 6 periods of kids, and what to do?
I'll be happy to take that. And I think, Susie, you can probably echo this. One of the ways that I've used the matrix in my classrooms, and you're going to see that in a minute. There's also a question about matrix in particular, like what that actually means. We're going to hit the matrix in just a few minutes and kind of walk through what it is you're going to do with that matrix, or how you can energize that with your lessons in your classroom. But kind of going back to your question, you can recycle it, you can repurpose it. So I've actually taken small plates and spread a little bit of matrix out on those plates for different groups to sit at their tables and work together, maybe in pairs or threes. And then at the end of the class, take what they found, the pics that they find, document them and document who finds them. But I've also thrown them back into the matrix again and done it again with the next class. That's one way. If you've got smaller groups, of course, you can have clear, concise pots of matrix that each team is working out of, and that's it. But in big groups, I had 175 kids. That's what I would do to kind of get everybody the opportunity that needs that.
Okay, and if we find something that, because we're gonna find something that I know nothing about, because this is not my field, which is one of the reasons I'm doing this, do you guys have any sort of a fossil guide for what we might find in there?
We do, and we're going to show you that momentarily as well.
Okay, thank you.
Great questions.
Thanks.
I did have a question about that. Sorry.
Go ahead, Cassie.
But you said that you're just going to get to the matrix in a bit, but do you suggest just looking through the matrix, or like microscopes or stereoscopes, or just
all of the above? You can use hand lenses, you can use dissection microscopes.
Okay.
Any of those are perfect. Those will all work. Even nothing at all, just the naked eye. You'll be able to find things that way as well.
Thank you.
I'm going to go back and just double check the chat box. Okay, there was a question about what do you mean by the matrix, the rubble with the fossils? Yes. And you're going to see how that rubble that you have, which we call matrix, how it came to be in that bag that you have. There's actually a whole process and setup that is entailed. So we'll go through that in just a moment. Let's see, Mary Lynn, can you explain again the geography of the site as it was years ago? Great question. And yes, do one of you all want to take that and just give kind of a brief overview again of the geology and geography of the site?
Yeah, so there's a really good website that John linked to that tells you all about the site. It tells you who was there, what was the environmental reconstruction, geographically relative to the ancient coastline where it might have been, and then some references. And I would say that's your touchstone where you can get all that kind of information that's written down for you. But just as an overview, this site is probably around between 5 and 6 million years old. It was closer to the coast back during that time than it is today. And we're digging up animals, many of which are now extinct. So that's a general overview, but the specifics of those sorts of things, as I said, are in the document that John linked for you in the chat.
Thank you.
Yeah,
And I see
and
John
we
also
don't
put
know
the
exactly how close it was to the coast, but you know, it might have been even as close as a couple of miles away from the ocean at that point, which is a lot closer than it is today.
I have a question about that. So because I have seen that site, but I saw it before when I was first applying for the participation, so I haven't seen it recently. I wondered, do you think that that site has enough information on the different types of organisms that my kids could potentially put together a food web there, or food chains, things like that?
Absolutely, yes. And there should be a fauna list in that article that John linked to, and the scientific names are there, and then the students could actually look up the scientific names and figure out whether they were a plant eater or a carnivore, and then they can reconstruct the food web, the ancient food web, using the kinds of animals that are listed in what's called in the fauna list there.
Can I share my screen real quick?
Listed there?
Yeah, is it possible to share my screen real quick?
Yeah, John, you should be able to.
All right, I'm going to share. Okay, so how do I do that? So hold on. Safari. Okay, it doesn't look like I can do it. I don't know if you all can. Sorry, I'm not.
Are you trying to share, John?
I just wanted to show the website. Do you want to show? Can you do that? Maybe.
Yeah.
I have a new laptop and I sometimes have trouble with sharing my screen for some reason. Yeah. So if you show the website.
I have a fauna list there. Do you have any idea of producers?
Yeah, so let me just show you something real quick.
So
if you click on, there's a fauna list. So if you click on mammals, for example, so click on mammals, and now a lot of this, there's not, they're colloquial names there, so you can kind of go and you could look up modern examples if you want, but some of them actually do have links that have a lot more information. So if you, for example, were to click on tapirus or here on Hydrotherium, which is a large extinct otter, and there's a lot more information about the animal if you scroll down. So we don't have this for all the species yet. We're kind of working on it, but there's a lot of information here. And so it isn't for all of them, but for some of them, you can go in and get a lot more information like this. But even for the ones that there isn't a lot of information, there is in parentheses for many of the things, whether it's mammals or fishes or reptiles or birds, because you can expand on all of those. In the parentheses, there's sort of a reference to some living analog, right? So the students could, Mitchell, I wonder if if it wasn't an amphisbaenid they found, but a glass lizard. Was it a glass lizard maybe?
Yeah, sorry, go ahead.
Anyway, sorry, I just saw that. But anyway, so, it says whistling duck, right? That's probably an extinct species of whistling duck. But if you look up modern whistling ducks, you can see how they fit in the ecosystem. So that could potentially help if you're looking at food webs. And yeah, it's a pretty extensive list of all the things found at Montbrook. You should be able to do something like that, I would think. And then some more detailed information about some of the species. Is that sort of along the lines what you were thinking? Yeah.
And Mitchell, did you have anything you wanted to add?
No, sorry, I was just going to say the Ophiurosaurus, the glass lizard, we've found from there before, but actually what we think was found this past year was an amphisbaenid, which would be a new record.
Cool. All right, so we will have plenty more time at the end for questions, as well as Stephanie and I will stay on for just a few minutes at the end of the workshop this evening. If you have any additional questions, we're happy to answer. But just to briefly go over our goals, of course, today we want to provide you with more content that you can bring back to your classroom. In just a few minutes, Mitchell's going to talk to us a little bit about his work, and that's going to be some of the bulk of the content today. And then Dr. McFadden or Bruce is going to talk to us about how some of those samples are prepped for you to use. So beyond that, like I said, we are going to get some background information on how things are taken from Montbrook and brought back to the lab so that they are ready to be analyzed and researched further. And then towards the end of the workshop today, we're going to start to brainstorm about, as you know, one of the requirements of this workshop is the implementation in your class. And again, that might be with the scientist. The scientist might come before that or after that. However you want to get that scientist involved is up to you. But at some point, you are going to have within this interim a scientist visit. So we're going to start to brainstorm ideas together as a group of how do you want to do that? Where does it make sense within your curriculum to involve that scientist, or just embed these concepts that you are learning through this workshop and start to get those juices flowing and those connections made, I should say. So that's really what we are here to do today. And without further ado, Mitchell, I'm going to turn things over to you.
Awesome. Thank you, Brian. Yeah, I'm just going to give a brief PowerPoint here that just kind of explains some of my research and actually a project that myself and Jeanette and a few of us have been working on that actually relates to the Montbrook site. So it's timely, convenient, it's ongoing. So it's exciting. I'm going to see if I can share my screen. Okay. All right. So yeah, my name is Mitchell Rigler. I'm a PhD student. I met, well, I was there this past Saturday, so I think I met everybody here. I am a student with John Block, and I am a vertebrate paleontologist here at the University of Florida in the Florida Museum of Natural History. And so just a brief intro on what I do, and I'll kind of transition to this to how you can apply it in your classrooms. But in a very technical, jargony way, what I do is I study, or I at least attempt to infer reptilian ecology in the fossil record utilizing high-resolution CT data and stabilized slope ratios as a proxy. Now, that's very jargony. So what it really means is understanding the lives of reptiles in the past is really what we're trying to do. All those terms essentially say nothing more than that. So first, what do I study? And also more specifically, what don't I study? There's a lot of preconceived notions. So paleontology, if you didn't know, it's the 200th anniversary of the coining of the term paleontology. So in 1822, the term paleontology in a French manuscript was first put forth. Roughly the etymology or the history of the word, it just means to study ancient life. Paleo is ancient, onto is life, and ology is study. So we study ancient life, and that sounds very vague and broad, and that's because it is quite broad. We do pretty much study anything that falls within that definition. So the tree of life, if you ever have seen this depiction, so this is the broad scale tree of life all the way, including bacteria, into plants, funguses, and into vertebrates, which is what specifically this group studies, vertebrate paleontology. But paleontologists and vertebrate paleontologists, botany, paleobotanists will study anything that falls within this tree of life over the 500 billion plus years of life on Earth's history. Sorry, my camera's in the way here. More specifically within the vertebrate tree of life, I study this group kind of generally. So the reptiles, so that includes things like lizards, which is a bulk of what I study, but that includes crocodiles, alligators included. I don't study, this comes as a shocker to a lot of people I talk to, anything related to dinosaurs. I've been on projects to look at mammals. A lot of people study, actually the history of Florida Museum paleontology has been mammal paleontology heavy, but I have never been or associated with anyone who does dinosaur paleontology. So paleontologists do not just study dinosaurs. I can't emphasize that enough. I teach a lot of undergrad courses, and they're almost even at that age, they're shocked to learn that paleontologists don't just study dinosaurs. In fact, I would say at this point, most do not study dinosaurs. In studying them, we use a lot of collections-based research. So at the Florida Museum specifically, we have a phenomenal collection. So we have the Vertebrate Paleontology Collection, which is here in the building I'm in right now, actually on campus. So we have the main exhibit hall, Powell Hall, which you've been to, the public display hall. We also have what's more of a research-oriented collection, which is in Dickinson Hall. And we have troves and troves of fossils all over the state of Florida. We have a herpetology collection, actually, which is what I use a lot of. So those are extant things. Those are things that are still living today, species that we find on Earth today that are still kept in collections. So we have that kind of material to look at. We have fossil sites all over Florida, Montbrook, which you all have been to, which is 5 to 5.5 million years old. We have older sites like Thomas Farm, the top right there, a site that's been studied for over 100 years, actually. That's from an even older time, about 18 million years. And then we have amazing technology the University of Florida has access to, like this in the very center there, CT scanning. CT scanning is a big component of a lot of research done in bird paleo here. Generally, I'm going to kind of go over just some of what I do. When I first came here, and the research I continue to do looks at squamates. So squamates are snakes and lizards. And one question I try and address is how squamates respond to climate change. And we use a model from the Paleocene Eocene. So that's a period that's just after the extinction of dinosaurs, about 55 million years ago. At that period of time, there was a massive global warming event that was comparable to what we see today. And we have a really good record of that actually in Wyoming specifically. So we have fossils from Wyoming that span this period of time, and we look at how animals responded to that climate change event. Specifically, I'm looking at how the lizards respond to that climate change event. So I have some fossils there. And we approach that through a number of different ways. Geochemistry is one way. That's what I show on the right. I'll explain a little bit more what geochemistry is and how I use it for projects that are more specific to Montbrook. I also apply the same methodology in questioning to Caribbean squamates. So again, the lizards and snakes of the Caribbean, a much more recent record. So these fossils that I look at here are not 55 million years, but they're actually more like 10,000 years. So quite a bit more recent, still old in any context that we normally think of, but very young for a paleontologist. So I look at how squamates respond to climate change over the last 10 to 12,000 years. And within that same period of time, we look at how lizards responded to the introduction of humans. So when humans arrived to these islands, how do they handle that massive transition? But more Florida specific, and I think more interesting compared to what you all are doing, is looking at how animals respond to Florida. So the geologic and the paleontologic history of Florida is actually really fascinating. So if we go, well, it is fascinating. If we go back millions of years ago, Florida is tiny. It may have been actually islands, actually. Sea level changes constantly. If we go back 18 to 20 million years ago, Florida could be a third of, half the size of it is today. If we go through time, sea levels fall a little bit, and we actually start to see Florida form more of a peninsular shape. We actually have a land exposure. As we move more recent, it gets bigger, actually so much so, we also have species that go extinct. I'll talk about this more in a second here, but with this change, there's a lot of biotic response too. But as we actually even move into the Ice Age, we actually see Florida even bigger than it is today. So Florida has geologically and geographically changed quite a bit, and animals have to respond and deal with that change. So going anywhere from half to a third the size to actually twice the size Florida is today, that represents quite a bit of change. And so how do animals respond to that? Sorry here. And yes, and we have a number of fossil sites. So actually in the bottom left corner, you'll actually see Montbrook. So this is actually what Florida would have looked like at the time of Montbrook. And there are other sites that are kind of within this age. We have other sites from more recent ages. So we have sites all throughout Florida. And one question that we had with all of these sites that we have recently begun addressing is the alligator record of these areas. So we have alligators. So we are the University of Florida, we are the Gators, and we have alligators from from all of these sites. They actually have a very extensive and rich fossil record. You all were at Montbrook. I think we found a few alligator bits, some osteoderms and some teeth. So you got to witness some of that. And we have these alligators from all over the place. So one inherent question then with something that we have such a good record of is how do they respond to all this change? How do they respond to geographic, ecological, or biotic change? And so that's actually a question that we begun 2 years ago, kind of in the midst of COVID, actually. And that's what I'm going to talk about right now, just briefly here. We approach this through a number of different means just to show you some of the tools that we use. One tool is CT scanning. So like I said on the right, so we do what's called 3D digitization. We digitize our fossils in 3 dimensions, and that allows us to analyze the fossils in 3D space. So on the right here is actually, it's a CT scanner. So not the CT scanner you think of in a hospital. This is an industrial CT scanner. We can put our specimens in there and get a nice 3D model of the outside and the inside. We can also, more recently, we have what's called a spider scanner or a light scanner on the left here. It actually just takes essentially thousands of photographs from multiple different angles and produces a very pretty surface model of the specimen. So we digitized our alligator collection. And just to show you what some of that looks like, this is a CT scan of one of our alligators. So here is the alligator skull. We CT scanned it, and actually it allows us to look at the outside and the inside. So in this red line here represents just a cross section through the skull. That's this top right portion, right? So we can actually see inside the skull by CT scanning it, which is phenomenal. We can also produce these really pretty surface models. So these are actually, I'm showing you 2D images, but these are actually 3-dimensional, beautiful surface scans of all these fossil alligators. Very rich record. We also did modern things. We looked at modern alligators and crocodilians. And we compared the things on the left. So everything to the left of that line represents an alligator from Montbrook. So from the site that you went. So these are 5.5 million year old alligator skulls. They are beautiful. They're amazingly preserved, well intact. And we have a bunch of these. Actually, we even have more since this was made. So we have more alligator skulls. And we compared that to everything to the right. And one question is the alligator at Montbrook a different species? One way that we can approach this as paleontologists is we look at the morphology. We look at the shape of the fossils, and we try and determine if the shape of that fossil is distinctly different from anything else that's been described. If so, that gives us an indication that's a new species. So we looked at shape change. This is a plot that essentially each one dot represents a skull, and any dot that's closer to another dot represents something that's very similar in shape. The further apart the dots are, the more different those shapes are. So we have some crocodiles on the right, we have caimans on the bottom, and actually we get a nice separation. So we have crocodiles bottom right, caimans bottom left, and then all alligators plotted in the center. And then actually within those alligators, we see nice separation. So this blue group represents the modern alligator species, that's Mississippiensis. And this orange group represents the Montbrook alligator. So that was a good sign. So we actually found an indication of morphological separation, morphological disparities between these two groups. We also took it one step further, and more recently, and Jeanette was the reason that this happened. So we thank her very much. Is the question of okay, what can we learn from something else that isn't extrinsic, maybe that's intrinsic? And in this case, the isotope chemistry of fossils actually tells us something about what they ate. So you are what you eat, plus or minus 1 or 2 parts per mil is a joke that we use in geochemistry, which says your chemistry is a product of what you ate. And so when you eat it, when you drink, that becomes your physical self. And we can measure that chemistry with what's called an isotope ratio mass spectrometer. So a mass spectrometer pictured here on the left essentially measures the different isotopes. It's actually, in case you're curious, a giant magnet. So on the right here is just a simple illustration. So we measure from the source, that source runs through a giant magnet and separates things into their different weights. That's all it is. It's really a big fancy magnet. And it allows us to tell us generally what was something eating, and also kind of generally what was it drinking, or what water source was it around. So we use this to try and tell us something about the environment or the ecology of the alligator. So what did we find? So again, we have massive climatic change these alligators are dealing with from all these sites. So we took all of the alligators from all these sites and sampled them geochemically. What did that result? Everything to the left of that -5 line that you see right there. So we've got 2 actual plots, carbon and oxygen, which are the 2 chemical elements that we're looking at. Everything to the left of that line tells us the animal is eating in prey that's freshwater source. So you're talking about trophic structure and food webs earlier. So everything to the left of that line represents an alligator that is eating more freshwater things, turtles, deer, something that came too close to the bank but isn't a freshwater food source. And all of a sudden, we actually started finding animals now that for the first time ever, we're plotting to the right of that line. We actually find populations of alligators that are distinctively plotting to the right of this group. So we actually have environmental change driving what's generally thought to be competitive exclusion, because what's happening is all populations before 7 million here, let me go back here, all populations before this window right here, when crocodiles go extinct, those are the ones plotting to the left of this line. When the crocodiles go extinct, that's when we start seeing them plot to the other side. So we actually have crocodiles scaring away the alligators until they go extinct. And at that point, they start diversifying, eating more marine source prey. So what's the grand conclusion? What is one of our main takeaways that I think you will be interested in is that because of its ecology and because of its morphology, the shape, we actually feel confident now that we have a new species. And this name was actually just recently decided upon by some of the people here as alligator pugnatus. This paper is in the works, so hush hush on this. No one else knows about it. But the name of this species will be alligator pugnatus, which roughly translates to the fierce or fighting gator. Pugnatus means fighting, and this is actually hearkening in reference to the early 90s and actually potentially even the 80s retro alligators. We're going to name this as an honorary for the University of Florida. So it'll be from the Montbrook site. All 4 of these represent the Montbrook alligator. It represents a new species. And so any of the alligators material that you might find, just know that represents a novel species. Last thing I'll say is how can this connect to maybe some of the learning standards? So I base these off learning standards that I'm familiar with. I did elementary school last year. So obviously, some of the innate questions that come with this, raising questions about the natural world. How did things live? Where did they live? What were they eating? Those are questions that students can ask, and those are actually questions that I ask every day. Using the 5 senses, don't taste, but please certainly touch, look, observe any shape that you might notice. We do put fossils to our tongue on some occasions just to see if it's bone or rock, but in this instance, that probably won't be very helpful. Ask how do you know? Why do you know? These are very appropriate questions, and these are things that we all ask constantly. Keep records of investigation. So when you see things, take notes. I think it would be great for the students to certainly keep record of what they maybe found or didn't find. We make a lot of inferences based on observations. So the concept of inferring and making physical observation. Recognizing and explaining, this is something that's important for, I think, any age, is that the scientific investigative process doesn't always necessarily parallel the natural steps of the scientific method. We always want it to be reputable, and we always want to make sure that's consistent. But sometimes science is truly an unexpected happenstance, like the isotopes with the alligators. We never intended on doing that until Jeanette brought up the idea, and it was an amazing happenstance. And sorting objects by observable properties, actually, and that can go into this next. Ooh, I have paused my screen sharing is paused.
It's still sharing, Mitchell.
Okay, sorry, my screen is doing something weird. Okay. All right, and then this kind of segues into when you're picking and sorting your fossils, some questions that people I think will have is how can you identify fossils from rocks? This is something you'll pick up on, but geometries certainly play a big part of this. If they're, geometries are usually a product of an organism. Rocks typically don't make consistent symmetrical geometries. So one thing you can look for. Colorations are another giveaway for certain fossils. So learning morphology and shape change. Sorting objects by their observable properties, so color, shape, and obviously looking for things of different sizes and how that correlates to what the different animals might be. So those are some of the general things I would think that could immediately segue into your learning standards. So I'm going to stop sharing here actually. And if anyone has any questions, feel free.
Mitchell, I did get a question, I think that was actually private chatted to me, but I think it was surrounding whether or not some of the slides that you showed that weren't, proprietary, whether some of the slides could be shared with the teachers.
Sure. Yeah, absolutely. I'll just send it to Brian, actually.
That'd be great. And we'll stick that information in your Google folders, teachers. We'll briefly show where that is once again tonight, just so you know where to find not only that, but also the different identification guides we mentioned earlier that Alicia asked about, and also some videos and images from Montbrook with your dig that were taken by Kristen when she was there with us. So Brian, do you want to do Q&A right now, or should we plug on and have Susie do her part?
I guess for the sake of time, let's keep going, and then we'll stay on if there are questions at the end.
Perfect. Susie, I want to pass it over to you.
Okay, so you just wanted me to talk about my experience with the matrix, correct?
That is correct. Just to kind of say as a veteran teacher, how this kind of rolled out in your classroom?
Okay, so, and just to kind of piggyback on one of the questions earlier about splitting up the matrix, like a small amount goes a long way. Like the kids will look through it a few times and it takes them a while. And I think also if someone else looks through it again, it doesn't hurt because they can miss little things. So it does take them a while to go through that bag, even though it may not look like a lot. So I had provided the students with small little paintbrushes, tweezers, and magnifying lenses, and that helped them to, you know, move around these little parts. We put in groups, I put like, you know, a small amount of matrix on a plate, and then they had like the little vials to put what they found inside of them. Once they had things in the vials, then we kind of had some plates labeled with like mouth plates and gar scales and vertebrae, and then they would separate them out into what you see here in these pictures. Once they started recognizing the shapes and getting it, like they were just finding them left and right. It was hard for us to really, like we had the identification guides, but it was hard for us to really, you know, identify things further than, you know, this is a vertebrae and this is, you know, a tooth. Like we didn't know exactly, you know, what kind of animal it came from, but we could just, they got really good at identifying, you know, what you see in the pictures. And then the things that we weren't really sure where it fit in, then we had Bruce come in and do a virtual meet with us, and he helped us to, you know, talk us through some of these things and tell us what we had that we weren't sure about. So that was really awesome, and the kids really enjoyed that. I also did have a microscope that connected to my computer, so we could look at the details of some of these things, and the kids really enjoyed that. They would ask me to sort through more matrix when we had like extra study hall time. They were so into this, and they would find, like, you can see that little tooth there that is just the tiniest little thing. And I was like, how did you even see that? But they wanted to look through more. They were so excited at anything that they could find. And so they really enjoyed it.
Susie, if you were to do it again, is there something you would do differently or a recommendation you would have for these teachers doing it for the first time?
It really actually went pretty smoothly, just having the tools ready for them. And, you know, I kind of improvised at times with, like, once they had their vial filled, they, you know, separate them out all together so we could see all the gar scales together, all the vertebrae together. But it really worked out pretty well.
That's excellent. Yeah, I mean, the pictures that you've taken here, the fact that you have something to show size is great. And I think it's really nice that they were able to kind of sort and classify things here. And as you get better and better, you can start to kind of whittle down to, you know, the different organisms that they might come from. And it takes a little practice, but I think that the scientists are there to help with too. So that's awesome.
What level do I teach? I teach 7th grade science. And someone asked what were the tools again. So I had like small little paintbrushes so they can move the fossils around. Then we had like little tweezers and magnifying lenses.
And I believe in your kits that you went home with, there were some little Petri dishes. You can split those dishes up so that different groups have a top end or a bottom end of the Petri plate. There's a few tweezers in that bag too, just to kind of get you started. But those are great tools to use just to kind of sort and push the sediment away from the actual, like little rocks away from the actual things that look like they're to be fossils. And they'll be able to figure it out and distinguish sooner or later. It's pretty cool.
Yeah, they picked up on it, you know, pretty quickly, starting to identify things. And then how long did I allow for sorting? It was really a whole class period. Like it took a while for sorting. And then, like I said, they wanted to do this more like when we had extra time. So we sorted a few times, but the main one was like, it took a whole class period to do some good sorting.
John has his hand raised.
Yeah, I just
Good, John.
wanted to make a quick point also with the matrix that you all were sent this time, which there is a little bit of a difference between this time and last time, because it worked so well. You guys the teachers did such a great job with their students picking the matrix. We actually sent you all home with what I suspect may be a much more concentrated bags of fossils. So there should be a lot of stuff in there. So these were actually interesting. You see how many fossils are there? That was actually from our spoil pile. So if you all looked on the side of the site where we were dumping our buckets, we just picked up that matrix and washed that through. And that's what we sent out with the teachers last year. And it produced all of those fossils. So all those buckets we were dumping had all these amazing fish fossils in it. Well, this time we sent you home with matrix that wasn't thrown in the spoil piles. This was matrix we specifically targeted because we felt it would be a high concentration in microvertebrates. So it's hard to say till you go through it, but my guess is there'll be a lot of shark's teeth, even more than what you see here, and maybe some other neat stuff too. So anyway, I just wanted to make that point. It's you really are part of the discovery, and you can definitely enforce that with the students that we're very interested to see what comes out of these bags, and more than happy to jump in and see what treasures are found for sure.
It's really helpful to have one of you guys come in virtually or in person, like after they sort through it, because then they really, got a lot more information out of what they actually found, because it was hard for us to identify everything.
Sure. And that can lead to, as John kind of alluded to before, you can start looking at those different species and investigating and looking at the life of that organism and what it would have been like ecology wise in that environment back then. So I think those are all really great extensions to what you can do in the classroom. Thank you for that very much, Susie.
No problem.
In the interest of time, I'm going to go ahead and move through. Bruce, if you'll tell us about the process of prepping that matrix that John just alluded to that is really fossiliferous. Can you quickly run us through what that
Yeah, well, because we're running short on time. So you've all been to the site and you all know that there are sacks around that we bag up matrix, as John mentioned, and we have lots of stock of matrix. Every one of those bags is research grade matrix, but it has the sand and then the fossils in the sand, so it's not concentrated. So what we do, what some of the technicians do in the museum is they wash the matrix, which is probably 95% sand, and the sand swooshes out of the screen, and what you have is the tiny fossils that are concentrated, and that's what we call the matrix. It's the concentrated stuff that we've brought from the bags, where we've already washed out the sand and reduced the volume to concentrate the bones and the teeth and some of the tiny stone particles. And this, what you see here, is what we refer to as the concentrate or the fossil matrix. And that's how we get it from the site. If we had just sent you home with bags of sand from the site, and you pick that, it wouldn't be as concentrated. So what we've done for you is really made this a rich potential for your students and yourselves to sort through every one of those grains and find the tiny fossils, just like Susan did with her class last year. Stephanie, is that the last slide?
This is the last one, I believe.
Okay, and then here's Rachel finding some vertebrae of tiny fossils, and these are the kinds of things that you'll find. When you take pictures of things, you'll want to take pictures. Always put a scale in it. And I think we probably didn't we give them we gave them some of the fossil rulers. A little bit of propaganda here. This is an online project we have called My Fossil, where you can actually upload pictures of your fossils that you find if you want to, but that's a whole nother story. Take any pictures you take, just get in the routine of putting a slide a scale next to them, so we have a sense of the size of the, because in addition to the shape that Mitchell mentioned, the size is really important. Okay? All right, I think if that's the last slide, because I know we're running short.
It is, and
Okay.
thank you for that. So we have in this presentation, which will be in your Google folder, just some standards that we've selected, that we highlighted, that we thought were, pertinent, from elementary to middle to, oops, sorry about that, high school, crazy mouse. So you can kind of sort through, find standards within, and you might even find others that are outside of this box. That's completely fine, whatever works for you. So we wanted to just quickly go over the overall agenda from here. As you know, we are looking at the content in the matrix today, kind of getting you set up for how to use that in your classrooms. We have another check-in on March 10th that we hope that you'll have progressed with how you want to do that implementation. And then one final virtual check-in on the 21st of April. Again, a place to ask questions, look at materials, brainstorm as a group, reflect as a group. And then finally on May 7th is our in-person. We're going to bring you back to the Florida Museum of Natural History this time to kind of see how it all wraps up into a pretty package. Once those collections have happened, we'll look at how things are prepared, and then how they ultimately might end up in exhibit in a museum and then shown to the public, so that we can educate others about all of the great things that are happening at these sites and the research that happens behind the scenes. Do you have a question?
No, not a question, a comment. So the other thing is, we're very excited that you're coming back at the end of this PD, because last year we didn't have that, and we wanted the teachers to mail the fossils back, and that was a challenge. So you're going to come back, and to finish off your participation in this project, you'll come back on May 7th. But the cool extra thing that you don't know about is that there's going to be a new fossil exhibit that will be open in the museum on that day. And you'll be able to go in and see people working in a paleontology exhibit called Fantastic Fossils. So you're going to we're not going to just sit around a table all day and have you talk about the matrix and develop and you're going to present your lesson plans, but we're also going to let you go back into the exhibits museum and participate, or at least take in this fabulous new exhibit that we're developing in-house. And John, do you want to sort of mention anything more about that? We're all very excited about this exhibit, but you all are going to have the opportunity to have lived being a paleontologist along with your class, and you'll get more out of this visit because of this experience you've had so far when you go and look at the Fantastic Fossils exhibit on the 7th in our museum. John, any other comments before we wrap up?
Well, just on coming back to that exhibit, I'm super excited about it also, and so I can't wait to show you all it. It's very interactive. We hope it's going to be very interactive. We're going to have a live lab set up processing fossils from Montbrook. We'll have video. We're hoping to do live from the field kind of broadcasts, or live from other places too, from teachers. I mean, there's all kinds of opportunities there. I'm also hoping to show some of the video that we did with you all in that exhibit space. So hopefully you'll be TV stars also when that's going on. But no, that's about it. I'm excited to help identify fossils as you all find them, as Bruce was saying as well. So hopefully I get to engage with you all that way. Mitchell, that was a great overview of the alligator research. That was cool. Thanks for doing that.
Yeah, of course.
Thank you, y'all. So one other little thing to add in reference to the May 7th date, we are going to showcase kind of what you did in the classroom. And so take good pictures and whatnot. We're going to just look at those expectations again, these 2 remaining workshop sessions that are virtual, the in-person on May 7th. And we're hoping you can kind of show us some artifacts of something that you did with regard to the Montbrook site and the different ways you've kind of tied it into your curriculum. So be thinking about that. And again, on this lesson implementation, please don't hesitate to reach out to us to schedule a scientist visit. We will be happy to do that for you, and take pictures if you're able. Certainly there is the need to have releases for any students that are in front facial pictures. And if you can make sure that happens, that's great. Otherwise, be discreet. And I think you know how to do that as teachers. So we again want to just have that information put into the site. And I think that's it on our PowerPoint. So I just really quickly wanted to show you the folder. In your teacher folder, there are the reference tools we were mentioning. There's a couple really good videos and all the pictures in here. And that there's a link that was sent to you all in email. So if you don't see that, let us know, Brian or myself, and we will make sure we get that out to you. And otherwise, we'll stay on for questions, but we're just about hitting 8 o'clock, so we don't want to keep you much longer. We will take plenty of time to reflect, brainstorm together as groups in these next sessions. And again, if you want to stay along with us here now, we're happy to answer questions or talk further with you.
I have a question, actually, if that's okay. So how would I go about getting a scientist? Just because I'm currently for marine science, I am entering we're learning about, fish fossils actually right now. So for chapter 10, so it has all the bony fishes, the cartilaginous fishes. So I was trying to implement it hopefully within, the next 2 weeks. I'm so sorry. This cat is just awful. She's just awful. Trying to implement it into the lesson within the next 2 weeks. So it would be rather soon. And I don't know if that's possible.
That's not a problem. If
Okay.
you can shoot us a request form immediately, we'll reach out to scientists and see if we can get something worked out for you. It might be right at the end or the tail end of that 2-week period, but we'll do our best to make that happen for you.
Okay, I appreciate it. And where's the request form, did you say? Oh, right there. I see it. Someone just
Brian,
messaged.
yeah, you can just put it in the
Okay.
chat box.
Perfect. All right. Thank you so much.
Of course. Anybody else have questions?
Alicia,
Okay, thank you. Sorry, I had to unmute. My lights have gone off because as I haven't moved. So I see the Google folder. I got into that. So there's 3 documents there. Am I seeing what I should see?
That's all that's in there currently. We'll be putting PowerPoints in there from tonight. And feel free, teachers, put things in there for each other too. I know that was part of the reason why someone suggested we have a Google folder to drop materials. And that's a great place for you guys to, again, collaborate and share out, network.
Stephanie, did Kristen Grace share photos with you guys already?
He did. It's in that document that says videos and images. All of the photos that she took are in there.
All right.
I have another question. So the last couple of weeks, I've been kind of swamped with some marine biology trips that we've been taking. And I have already some ideas from tonight. Thank you for the matrix. I was wondering, I know Dr. McFadden, I had asked you about the legalities when we were on the trip or at the Montbrook site, and I understand the legalities. We have a really nice state park next to us, Oscar Scherer, and we also have Casperson Beach, really 5-7 miles away. I'm wondering, do you think if I approach the, local, like the state parks or the county who owns Oscar Scherer, runs Oscar Scherer, if they would allow our kids to take a field experience down there as long as we don't take stuff with us? Have you known anyone to do that?
yeah, I can't Stephanie would know better than I would. I would say every county could be different, and every municipality and state parks are really touchy about fossil collecting. So it really depends upon where you want to go and who's in charge of oversight for those. So I can't really, I don't have a blanket statement about the legality. It's really much more
I
complicated.
think that the beach is a well-known collecting spot for amateurs. So I think that is legal as I understand.
I believe so.
Yeah.
Wouldn't
It
you,
was,
park, I don't
up
think.
until recently, but I was telling Dr. McFadden that in the last couple of years, they put up signs. So I'll approach the county about the beach because I understand state parks are really, really touchy about that. And I wouldn't want them to disturb stuff at the state park, not knowing where to find anything, whereas Casperson is a very well-known place. It's practically all matrix. So,
Yeah, I saw a
thanks.
photo of people collecting there last weekend. It was full of people, so they must be doing it.
Okay,
Yeah,
thank you.
Casperson I've collected as well. I think you can do it there at the beach.
Yeah.
Sorry to jump in, but I take my students there every year on a field trip. The last 2 years is the only one I haven't. And it takes us almost 2 hours to get there from Tampa, and they love it. But you have to prepare them that they're not necessarily going to find as much shark teeth as they will. There's tons of stingray plates and also ice age bone fragments. So I talk a lot about predator-prey relationship because obviously sharks are eating the stingrays. And so you're finding both. but my students absolutely love it. And
Reese,
I've
would you
done-
be willing to put any plans or things that you do with them in that teacher folder? Because I'd hate to reinvent your wheel.
Yes, I have all kinds of stuff. I have handouts for them to ID the fossils. I've actually recreated my whole evolution unit using shark's teeth that I've gotten diving, but you can also get them from the beach. And that where they have to tell me like why some of the teeth are wider, some are more narrower. And we do a contest. The kid that finds the longest tooth wins $20. Like they love it. And they're on the bus for an hour and 45 minutes. Still love it.
Do you have them use the beach or the nature trail behind the beach?
No, the beach. And we don't talk to anybody. We literally just drive, and I have a bunch of sand collectors and I have them bring colanders from their kitchen, and we just go down and they absolutely love it. And we're there for like 2 hours, 3 hours, because you know we're coming from Tampa, and that's enough. Like you don't need a lot of time. And
Okay, thank you.
yeah, so it's fantastic.
Awesome, thanks.
Anybody else have questions? All right. Well, this is exciting stuff. I can't wait to see what you all do. And again, feel free to reach out to us.
Okay, thank you.
Absolutely.
Thank you, guys.
Good night, everyone.
night.